Good afternoon. My name is Michaela Williamson, and today I will be presenting over the article titled Endoplasmic Reticulum Stress, Major Player in Size-Dependent Inhibition of P-glycoprotein by Silver Nanoparticles in Multi-Drug-Resistant Breast Cancer Cells. Listed here are the authors, and I chose this paper because my grandmother was diagnosed with breast cancer a couple years ago, so immediately I just wanted to see what this article is about. And the main focus within the article is just to see how nanoparticles can affect the breast cancer cells within that are multi-drug resistant. First, I want to cover just what breast cancer is. 1 in 8 women are actually diagnosed with breast cancer, and it is estimated that within 2019, 268,600 cases will actually be diagnosed. And then it is also estimated that non-invasive breast cancer, within 2019, there will be 62,930 cases diagnosed. And that is just shown in this figure here based on the ages of women and the percentage of cases within those age groups. The most common types of breast cancer are going to be ductal carcinoma in situ, invasive ductal carcinoma, and invasive lobular carcinoma. Next, I want to cover what P-glycoprotein is, and in this paper they are going to refer to it as PGP. It is a multi-drug resistant protein, and it's also referred to as MDR or MDR1. It is a foreign substance remover within the plasma membrane. So here you can see that the plasma membrane is light green, and the P-glycoprotein is on the edge or centered right here. So it's going to remove the foreign substances from the cell. It is a MDR amplifier within cancer and also a chemotherapeutic inhibitor. Next, I'm going to cover what silver nanoparticles are, and this paper is going to refer to them as AGNPs. They induce neuron toxicity within the cytoskeleton component degradation, and also they cause mitochondrial dysfunction, which can lead to cell death. Real quick, I just wanted to talk what cytotoxicity means. It's just going to mean that whatever it is harmful or toxic to the cells and could potentially lead to cell death as well. And here I just showed a picture of the silver nanoparticles within different sizes, so they can have many different sizes that they range from. Next, I want to cover what multi-drug resistant or MDR cells are. They have an increased tolerance of oxidative stress, apoptosis, which we should know is cell death, activated DNA repair, and also modulated signal transduction. And there is an increase, especially when P-glycoprotein is overexpressed. And this figure is just showing that depending on if the cells are multi-drug resistant, there can be different targets, and the cells can have a different effect on that molecule. So the purpose and the part of the paper that I wanted to focus on was, can AGNPs induce cytotoxicity in breast cancer cells? And this was actually the first part of the results, and I thought it was the most important because without this they could not proceed to the following parts of the article. So the first method that they used was transmission electron microscopy, which is referred to as TEM. And this is the TEM device that's used. It's an electron microscope with a single atom column, and it shows an increased resolution of the sample that can contain organelles, viruses, and macromolecules. And in order for it to be used and get the proper results, it requires a vacuum environment. The next method that's going to be used within this section is dynamic light scattering, which is known as DLS. And it's also referred to as quasi-elastic light scattering, which is QELS. And here you can just see that you have a laser that's going to go through like a concave either mirror or lens through the sample that contains whatever you're trying to view or look at. And then the light is going to be scattered through the detector, and then based on that, it's going to show the particle sizing. So once the intensity fluctuates, it's going to count that and be able to tell what it's measuring. And it can measure nanoparticles, colloids, or just plain molecules. The other method that is going to be used is Western blot. So you're going to have a gel through a gel electrophoresis that contains your proteins. From there, you're going to transport or transfer those proteins to the PVDF membrane. You're going to block the non-specific sites that you don't want to see. Then you're going to use primary and secondary antibodies to either target the protein and then target the antigen. And then from there, you will see the detection of your proteins that you want, and you will be able to analyze them. The last method that was used was MTT assay, which is a colorimetric assay that is used for cellular metabolic activity. As you can see here, this is what it will look like once you're done with the MTT assay. And it reflects the viable cells present. So it shows how many are still there. So MTT is a yellow water-soluble tetrazolium dye, and once you put the DMSO detergent in with it, and it goes through all the steps, you will see the purple color, which is the formazan crystals that are formed. And then once you have this where it is purple, or you see purple, you measure the absorbance at 570 nanometers with the Synergy HTX microplate reader. So through all the methods, they were able to see that AGNPs actually do induce size-dependent cytotoxicity in breast cancer cells. The silver nanoparticles were verified via TEM, and here, these were the results from TEM. You can see the 5 nanometer and the 75, because those were the two sizes of the silver nanoparticles used. And then these graphs correspond with what they were able to see in these figures, and it just shows this is the size on the X, and then the number of particles for each of the sizes on the Y-axis. And then also DLS shows the size distributions assessed, to where silver nanoparticles were applied to both the multi-drug resistant cell lines and the multi-drug sensitive cell lines, and those are going to be shown here. Where were the silver nanoparticles present? Both of the different cells took up the silver nanoparticles, but the 5 nanometer particles were shown in the membrane-coated bodies within the cell. The 75 nanometer particles were shown only in the cytoplasm, and no silver nanoparticles were found in the nuclei, mitochondria, or the endoplasmic reticulum. The MDR phenotype of the MCF7 KCR was verified, and that is going to be the drug-resistant cell line. And that was verified through the Western blot. So you can see that the P-glycoprotein is shown here within the drug-resistant cell line, but not in the drug-sensitive. So there was massive expression of that P-glycoprotein. But the MCF7, so the sensitive cell line, lacked the ABC transporter, which is the ATP binding cassette. And then the efflux activity was confirmed in this graph here. Cytotoxicity of cells was determined, and that was through the MTT assay that I had mentioned in the methods. And with this, it's going to detect the toxicity of the cells through the nanoparticle size, within the treatment time, and also the different cell types that were used. It was determined that the more cytotoxic were the 5 nanometer silver nanoparticles. And as you can see here, that the numbers are lower, showing there are less cells valuable than the 75 nanometer. So the 75 nanometer silver nanoparticles were less cytotoxic than the 5. So the silver nanoparticles induced cell-dependent cytotoxicity in breast cancer cells, and the 48-hour treatments were more effective in the cells. The MCF7 cells had increased sensitivity compared to the drug-resistant, and that was in both of the different sizes of silver nanoparticles. And the drug-resistant was efficiency defeated in both 5 and 75 nanometers. So this study concluded that the larger silver nanoparticles did modulate P-glycoprotein activity and synthesize MDR in breast cancer cells. And then the rest of the study showed that ER stress was a favorable target for multi-drug resistant cells, and that silver nanoparticles were useful agents. Some future research that I think should be looked at is, we looked at 2 different sizes of silver nanoparticles, but what other sizes could be used? Are any going to be too large? If they're larger, are they going to be more harmful? I just think that more should be focused in on this to see exactly what would be too much, and maybe even what other types of cancer cells could this benefit. I know when I was looking at my different papers and researching this topic, Leukemia was brought up, but I didn't know if there were any others, and I think that other types of cancer should be explored too. So I think that should be some future research done. And here is my reference. Thank you.